The overall aim of this procedure is to evaluate the ability of a molecule to elicit chemotaxis and other migratory responses in explanted trunk neural crest cells. This is accomplished by first isolating trunk-level neural tubes between roughly 8 and 15 somites in length, and culturing each of the neural tubes overnight on separate fibronectin-coated cover slips to allow for neural crest emigration. Next, an optimal neural crest culture is selected. The neural tube is removed from the culture, and the cover slip containing the culture is mounted onto a modified Zigmond chamber so that the straightest border of the culture is parallel to the vector of the molecular gradient to be applied across the culture. The third step is to generate a molecular gradient across the culture and then film the migration of the peripheral neural crest cells along the previously selected straight border of the culture. The final step is to track and analyze the migration of the peripheral neural crest cells positioned along the selected border of the culture using ImageJ software. Ultimately, it can be determined whether the selected molecule can alter cell directionality or other migratory characteristics, such as speed, through the analysis of the cell trajectory data obtained.
The main advantage of this technique over existing techniques, like the Boyden chamber assay, is that at a very low cost, this method can be used to analyze the migration of already polarized cells on the periphery of primary explant cultures using time-lapse imaging.
After incubating chick eggs for 56